Hello and welcome all in this video. So we are going to discuss about sorting and identification of fish seed of desirable species. So the spawn identification is possible only with the aid of microscopic examination. However, sieving of spawn using selective sieve helps in segregation of spawn of the desirable species, that is the four Indian major carps from the undesirable ones, because we can collect or we can sort or we can have to identify those fish species seeds of those species which are culturable, or we have to culture that particular fish seed. So regarding the hatchling and fry, which are visual identification is not possible unless 3 centimeter size has been attained of that particular fry. Then below this size, identification of fry of desirable species against those of undesirable ones can be made only under microscopic examination. So at the time of segregation and identification of fish seed from relevant sources, we have to some steps or some examination test by which we can identify or segregate or sort that particular species, eggs of that particular species which are desirable species. That although collection of fish seed is very economical when taken from relevant sources, it presents in a great difficulty for identification and segregation of fish seeds for a particular desirable species from the mixed lot. It is only seldom that there are waves of pure fish seed in rivers. Stocking of mixed lot is surely a great risk, since it may turn out highly uneconomical and wasteful if there were a preponderance of undesirable species in the lot. So there are some steps, there are ways, there are a number of ways, particularly there are three steps or three types by which we can identify the spawn and fry of desirable species. There are a number of ways to identify spawn and fry of desirable species. In there are three important steps or three important techniques. With the help of that, we can identify or we can segregate these particular spawns and fry of desirable species. In that first method, that is called as visual identification. Visual identification is possible and easy with advanced fry, but difficult or impossible with early fry or spawn. There are some common criteria used, include such characters as there are some criteria by which we can identify or sort that particular fries of particular species which are required in the fish culture method. So in that, first is the layer of water occupied. Some species prefer the surface while others stay near the bottom. Where this pond is occupied, in which part of the water column they can found, that is very important to identify, or that is a method of visual identification of the fry or fingerlings, fry, particular fry and spawn. That is layer of water occupied. Some species prefer the surface while others stay near the bottom. Manner of swimming. Observe how they move and if they are schooling. With the help of that manner of swimming, we can also identify the particular fish spawns or fry. Body proportion. Compare the size and shape of different species. With the help of that, we can also segregate the fish seed. Size of eyes. Note the relative size of the eyes, which this is also an identification key to differentiate the fish species. Shape, size and situation of fins. Look for distinctive fin shapes and placements. This is also a key to identify the fry and spawn. Presence of color is also important. Observe any unique coloration patterns that is also unique for each and every species of the fish. These are some methods that is called as visual identification. With the help of that, we can identify, we can sort the fish species, particularly prawn, that is a particularly spawn and fry of that species. Second method, identification with the aid of microscopical examination. This is applicable to both spawn and fry and highly useful in determining the quality of spawn. The common characters include shape and size of yolk sac. These are the methods which are the steps or stages or methods with the help of we can sort that particular species, that is called as microscopical examination. These are some characteristics with the help of that microscopic examination we can also sort these particular species, spawns and fries. Shape and size of the yolk sac, presence or absence of oil globule, segmented unsegmented nature of yolk, situation of anal opening in the body, nature of fin fold, rudiments of fins, pigmentation of eye, opening of mouth, presence or absence of barbels, etc. These are some characters by which we can identify the particular fish species, spawn and fry of particular fish species. Third method, which is called as Chinese method, that is the identification with the aid of differential dissolved oxygen requirement. This is popularly called as Chinese method, found highly useful for Chinese carps. The criteria include a different degree of demand of dissolved oxygen by different species at hatchlings or fry stages, as manifested by accretion of different layers of water from surface through subsurface and middle layer to the bottom layer by the fry of different species according to their oxygen requirement when the mixed fish seed is forced to crowding, resulting in severe depletion in available dissolved oxygen. With the help of or with the depletion of the oxygen demand, we can also identify the particular type of fish fry. Particularly, this method useful for identification of Chinese carps. Out of that three methods, the first two methods are of little application for commercial usage in segregation of spawn or fry. The Chinese method useful only when the mixture consists of fish seed of fry of different stages from spawn to early fry is highly practical and finds us for commercial applications. So this is all about the segregation or identification of fish seed. In our next video, we will discuss about the transportation of fish seed. Thank you.